So welcome back to my Biotechnology classroom. And today we discuss a very common experiment. This is something that anybody who does biotechnology cannot escape, because if you're doing biotechnology, you would at some stage do a polymerase chain reaction. And one of the key important features of polymerase chain reaction is the designing of primers, because ultimately your wet lab experiment will depend on how specifically you've designed the primer and how correctly you've designed the primer. Right. So in today's lecture, we'll discuss how to retrieve the sequence specifically for primer designing and what all precautions you must take while retrieving this sequence. And in the next part of the talk, we'll talk about how to design primers using primer 3. Let's first have some basic idea of what is the requirement for designing the primers. Right. So this is an example here. This is your longer context of the DNA. Let's say this is genomic DNA. And the 5' end is represented by the blunt end here. The 3' end is represented by the arrowhead here, right? And here is our gene of interest. This is the gene that we want to amplify. What should be happening is that you have a primer here and you have a primer here at the start and end of the gene, and that should be able to amplify your gene of interest, right? But the problem is that you also require certain properties or prerequisites that a primer must satisfy, right? And those prerequisites sometimes may not be satisfied by this start and end of the gene sequences. So therefore, what can also happen is that instead of having a primer exactly here and here, you may have them slightly outside of the gene if you want to amplify the complete gene. So let's look into the properties of the primer that are required. So first, of course, the important property is that the primer has to be unique, right? So the primer binding sequence must come once and only once in the entire genome that you're looking at, or the entire larger DNA that you're looking at. You cannot have the same complementary sequence elsewhere on the DNA. In this context, what is also important is that the primer should not be designed to a repeat sequence. So as the name indicates, repeat sequences will be having multiple copies in the DNA, and therefore, if you design a primer by mistake to a repeat sequence, the primer would go to multiple places in the DNA and give you non-specific amplification, right? So therefore, you have to ensure that the primer is not designed to a repeat sequence, and also ensure that the primer sequence is long enough to be coming just once in the entire larger DNA that you're using for your amplification. Right? Then the primer composition is important. It must have around 50 to 60% GC content. Remember, GC means triple hydrogen bonds, and therefore the hybridization is stronger. And at the temperature that you want to hybridize, around 55 to 65, a high GC content would allow you to hybridize more stably, right? So this is also important. And if you talk of primer composition, if this primer is going to attach here on the strand here, then this primer has exactly the same composition as the top strand, right? So therefore, the primer composition itself depends again on the composition of the region where it is going to anneal. Sometimes if you design a primer here, the sequence composition may not satisfy these conditions. Right? So this again is context dependent. Sometimes you may not have a sequence composition of the gene that allows you to have a primer exactly at the start and the end of the gene, right? The TM, which is an important criteria for specific amplification, even if you have a very specific primer, which is a unique sequence, it may not show you specific amplification if the melting temperature of the primer is less than 55 degrees centigrade. So in fact, if you want a non-specific annealing with a specific primer, you can bring down the temperature of annealing, and that would allow you a non-specific amplification as well, right? So therefore, again, the TM of the primer is important. It must be between 55 to 65 degrees centigrade. Again, the TM of the primer is dependent on the composition of the primer. A general formula that we use, and which gives you an approximation of what is the TM, is 4 into G plus C plus 2 into A plus T. And sometimes, again, if you design a primer here, this may not satisfy the TM considerations. And therefore, again, you may have to move your primer a little outside, right? And likewise here, you may have to move this primer a little outside. Now, the challenge in primer designing is to how close you can design a primer to your target, right? So that is where the primer designing comes in. You also need to avoid dimer or hairpin loop formation. So one may argue that if you want to increase the TM, you could increase the length of the primer, but there are reasonable restrictions to how long a primer can be. And if you see here, I mentioned that for a specific primer, it should be not less than 18 bases, because the lower the length of the primer, the more the chance of random occurrence of that complementary sequence on the genomic DNA, right? And ideal cases, it should not be more than 27 bases, because more than 27 bases, you could have dimer formation. You could also have formation of hairpin loop. Here you are the complications that may arise if you have too long primer sequence. One, you could form an hairpin loop. This is basically the primer bending on itself and finding regions of complementarity. Second, you could have a self-dimer where a pair of our primers hybridize to each other, and then your PCR will effectively happen here, thus serving as template. The 3' end of the primer will get extended here. Likewise, this one would serve as template and the primer would get extended here, right? So this results in a dimer, self-dimer. You could also have a heterodimer where the forward and reverse primers complement each other in certain sections. So therefore, now if you look at the points one more time, so many a times, instead of having absolutely ideal primer pairs amplifying exactly your target of interest, you may actually have to design primers slightly away from your start and end of the gene to get the amplification. This primer pair would be your primer of interest that satisfies all the conditions given here. So now let's have a context of things. So the first thing that you require is your sequence. So most of the times when you're designing primers, you basically pull out or retrieve the sequence with extra flanking DNA sequence here. So for example, here, if your gene of interest has coordinates of X and Y, let's say on chromosome 1, so you retrieve the sequence with X minus 500 and Y plus 500. You retrieve this sequence here, and your search space is now, if you're amplifying the gene of interest, obviously your primers have to be outside the gene of interest. So you are looking for designing a forward primer in the 5' flanking sequence here. This is this one. And you're looking to design the reverse primer in the 3' flanking sequence of your gene of interest, right? So you need to retrieve the sequence for primer designing with certain specifications. Number one, retrieve sequence with the extra flanking sequence. This is your 5' flank, this is your 3' flank, and we're talking in the context of the plus strand now. Then you also have to ensure that this sequence is retrieved in hard mask, which means if there are any repeat sequences in these flanks, they are actually converted to capital N, right? So that the primer design software is blind to these sequences and does not design primers to repeat sequences. So retrieve sequences in hard mask, repeat nucleotides converted to capital N ensures that primers are not designed to repeat. Then if the gene is on the plus strand, you can directly retrieve the sequence. So if the gene is on the plus strand, which is also the sequence in the database, you can directly retrieve the sequence. But if the gene is on the minus strand, then remember the gene orientation is 5' this side and 3' this side. So while the coordinates are still the same, the gene has to be pulled out in the reverse complement fashion. So here you are. If the gene is on the minus strand, retrieve the sequence after reverse complementing. As for the other strand, the 5' 3' orientation is off. Gene always begins in the 5' end. So this is important that if your gene of interest is in the minus strand, you need to do a reverse complementation of the sequence to get the final gene sequence, right? Once you have the gene sequence, we'll use Primer 3 software to design the primers, right? So this is the basics of primer designing. Then we move on to retrieving the gene sequence. And let's say our test case is GAPDH gene in the human genome, right? In the first step, we'll first retrieve the sequence using the UCSC Genome Browser. And in the second step, we'll design the primers using Primer 3, right? So now let's go back there. So we Google out for UCSC Genome Browser, the Asia mirror site. Here we are, and this will take us to the first hit, and you could directly land on the home page of Genome Browser Gateway. So this brings us to the Genome Asia page of UCSC Genome Browser. The landing page leads us to human genome, and if you want to go to some other genome here, there is a whole list. You could simply click on the genome and it will lead you to that particular genome site, right? So here you are. If you see, if I want to go to chimpanzee, I could just click here and it will take me to the chimpanzee genome, right? We go back to the human genome here. And here, if you see, there are assemblies, and I'm sure you would know that the current assembly is the GRCS38 assembly. If you want to go back to previous assemblies, you could go back to your previous assemblies, which are archived here. Here you can search your gene of interest with the name of the gene, right? So let me type GAPDH, and here if you see, it gives you a list of all the entries that begin with GAPDH, and our interest is in the first gene here. So this is your GAPDH gene. I just click on this, and then of course I click on go. Right. So here we are on the annotation page. This is the GAPDH annotation page here. On the top panel here is mentioned the coordinates of the GAPDH gene. So this is chromosome 12. Starting from position 6534517 to 6538371 is the GAPDH gene. So these are your X and Y coordinates that I've shown you in the earlier slide. And then of course the gene is 3855 base pairs in length. And then of course, if you move down here on the chromosome 12 ideogram, the red line indicates the position we're looking at. This is our location of the gene. It is on the short arm or the P arm of the chromosome 12, right? And these are the various isoforms obtained from GAPDH. There are other annotation details which are not of our interest as of now, because we're more interested in the gene sequence here. Other important thing is that if you see, these arrows are pointing towards right direction, which means that the gene is on the plus strand. Now we go to retrieving the sequence. So for retrieving the sequence, you could go to view, and on view you click on DNA. This will take you to the DNA formatting page. So here you are. This is the position that you're looking at. This is the position of GAPDH gene. Remember, I told you for designing primers you need extra franking sequence. So you pull out extra 500 bases at the 5' end and another extra 500 bases at the 3' end. And then of course we come to sequence formatting options. All uppercase is fine. Because we want to get the hard mark sequence, we must check on mask repeats option. And then of course we mask repeats option. We want to hide the nucleotides corresponding to the repeat sequence. So we'll say mask repeats to capital N, right? So whatever nucleotides correspond to a repeat sequence, those will not be visible to the primer designing software. For primer designing are automatically excluded from primer design, right? We do not need to do a reverse complement here because our gene is on the plus strand, so we can directly retrieve the sequence. So we say get DNA here. Right? And we get our sequence in FASTA format. Remember, FASTA format, it will have a greater than sign followed by a one-line annotation of what is the sequence about, and then followed by the sequence itself without any indexing or numbering of the nucleotides. And then we move on to primer 3. So here you are. This is the primer 3 software. If you see here, this is the place where you can paste your sequence of interest for which you want to design the primers. And it also mentions that it will accept A, C, G, T, and N, and or any other alphabet will be treated as N again, right? So FASTA format is okay. So we will use the FASTA format. Please N out undesirable sequences. So it already tells you that you should ideally get your sequence in hard mask where the repeats are converted to capital N. If you're not doing then in that case you'll have to specify your mispriming library. So because we already have our sequence hard masked, so we will not specify any mispriming library or repeat library here. If we were not having our sequence in hard mask, then we could have basically specified which repeat library to use, and at the back end it would have again matched your sequence to those repeat libraries and masked those sequences. But in our case, we don't really require this, so we'll directly paste our sequence here. Now, there are two things that you need to specify. Because we want to amplify the entire gene, we cannot have a primer inside our gene of interest. Therefore, we need to specify to the software the region where primers are not to be designed. And that is given by this excluded region here, right? So if you see here in the excluded region, you could mention your start coordinate and the length downstream until where you do not want a primer to be designed. So in this case, we have retrieved the GAPDH gene sequences, 500 base pair extra flanking sequence on the 5' end. So in this case here, our gene starts at 501, right? And then, of course, the length of our gene is 3855 bases. So from 501 until the next 3855 bases, do not design the primer. This is the instruction you're giving to primer 3. So we say 3855. So if you see here, it is also mentioned in the note here that if you mention 401,7, forbids selection of primers with 7 bases starting from 401, right? So it is forbidding the selection of primers 3855 bases starting from 501. Now you have specified that your primer is not to be designed within the gene sequence, but we also run the risk of getting both primers designed to either of the flanks, right? And let me take you back to the slide here. So here we are, and what we have done till now is to retrieve the GAPDH gene sequence with 500 bases extra at the 5' end and 500 bases extra at the 3' end. Next, we also want to avoid both primers getting designed to one of the flanks. For example, here I show you this. It may happen because you've defined the primers are not to be designed within the gene sequence. It can happen that both primers get designed to your left flank or to the right flank, right? So we need to instruct the software very smartly that one primer you need to be designed at the left flank and the other primer you need to be designed to the right flank. And how do you do that? So if you see here, your gene size is 3855 bases. If both primers were to be designed to the left flank or to the right flank, the maximum size would be 500 bases because, the flank size is 500 bases. So if we could define our minimum product size of the gene as 3855, then you're automatically instructing the software that you design one primer in the left flank and the other primer on the right flank. So let's do that now, right? So here we are, and we will use this option here. If you see, this is your product size range. So we define our minimum product size as 3855, which is the size of the gene. And then we also need to give a bit of a range so that the primer can scan through the flanking region and find the best fitting primer. So let's say to begin with, we give a 200 base flexibility. So here is our option. We say 4055, right? And sometimes it may not be able to pick up the primers because of the conditions that you've mentioned here. The general primer picking conditions. Primer size 18 minimum, 20 optimum, maximum 27. So first the software will scan through the flanking sequence to find an optimum primer. If it doesn't find an optimum primer, then it will look for the maximum and minimum ranges. Primer TM minimum 57, optimum 60, maximum 63. GC content, we have given minimum of 20, max of 80, but preferable is between 50 to 60%. And we pick our primers, right? So if you see, we pick our primers here. Here you are. So this is the result space for primer 3 output. First, it says primer picking results for the AG38 DNA. So this is the def line of our sequence that we had given, and it has come exactly as that. Then if you see, it says no mispriming library was specified. This is because we already used a hard masked sequence, and therefore we did not use this to specify any repeat library. Then, of course, the indexing is 1 base. So the first nucleotide is number 1 here. And then, of course, you have the left and right primer details here. So if you see, the left primer starts at 353. The length is 20 bases. The TM is 59.93. GC content is around 55%. And then, of course, you have the sequence here, which is given here. Likewise, the right primer is starting at 4390. The length again is 20. The TM is compatible within 3 degrees centigrade of each other. GC content is 50 to 60%, so this is also fine. And then, of course, you have the primer sequence here, right? If you look into the context of the actual gene sequence that it given, so it says the total sequence size is 4855, and all of this region was included for the primer designing. And then, of course, the product size you get is 4038 bases. So 3855, and this is 4038 to 145 plus 38. roughly around 200 bases extra are getting amplified, right? The X indicates the start of the gene, the excluded region. So if you see, this is marked as X until the end of the gene, right? So this was your excluded region. And then the greater than and less than signs indicate your forward and reverse primers. So if you see here, this is the forward primer, and this is some distance away from the start of the gene. So now our effort would be to try and move it inside and more closer to the gene, right? And if you move down and scroll down to the right primer or the reverse primer, so this one is close by to the end of the gene and probably cannot be moved any further because it doesn't satisfy the conditions of the primer picking. So we have an option of trying to check out for bringing this primer closer to your target here. So we go back here and we redefine the product size range. So our minimum product size is 3855. We now give just 100 base flexibility here. So we say 3955. So now let's pick primers and see if the primer has moved in further or not. So here you are, and if you see now, this has definitely moved further inside. So this is now just about, the starting position is 462 and the length is 22. So 462 plus 22 is around 484, which means 16 bases extra would be here. Some 16 bases around would be extra here, but it has definitely moved closer to your start of the gene as compared to what it was earlier. It was earlier around 352 or something, right? So this is how you design your primers. And then of course, this remains at the position where it was earlier. And then if you go down, there are other 4 extra primer pairs that are given. It is basically roughly very close to the positions that are mentioned at the top, but 1 or 2 nucleotides here and there is what is mentioned here, right? So this is how you design specific primers for your amplification of a given piece of DNA, right? So we stop here and I'll see you again with another interesting exercise next time.